So, welcome students. Myself Satyabrat Sarma. I am Assistant Professor Assam Downtown University, Faculty of Pharmaceutical Science. Now we are going to discuss the third part of the drug excipients compatibility study, that is the analytical techniques. Okay, so let's begin with the analytical techniques. So now I am going to start the drug excipients interactions. So what are the analytical techniques we are using for detection of drug excipients interactions? So this is video is made for the MPharm first semester, uh, that is pharmaceutics. Okay. So in case of the analytical techniques, we are using various type of detection of drug excipients interaction parameters. Okay, we are using various analytical techniques. Okay, first one is the thermal method, second one is the spectroscopic techniques, third is the chromatography, then accelerated stability study, and miscellaneous. Okay, so in the thermal method we are using DSC, that is differential scaling calorimetry, and DTA, that is differential thermal analysis, isothermal microcalorimetry, and hot stage microscopy. So in spectroscopic techniques, that is FTIR spectroscopy, powder X-ray diffraction, then solid state NMR. Next, chromatography techniques, that is SIC self-interactive chromatography, TLC thin layer chromatography, and HPTLC. Then HPLC, that is high pressure liquid chromatography. Okay, but in our analytical purpose, we are basically give the importance DSC, DTA, and HPLC. Okay, and also FTR. So next come with the DSC studies. So differential calorimetry is the one of the most essential and useful analytical method to get the drug excipients compatibility parameters. Okay, so it is mainly based on the thermal changes of the thermal properties of the excipients or any chemicals. Okay, so what is the method? The pre-formulation scanning of drug excipients interactions required 1:1. That means the ratio should be, drug and excipients ratio should be 1:1. Okay, to maximize the likelihood of the observing an interaction, mixture should be examined under N2 to eliminate oxidative and pyrolytic effects at heating rate 2,5 or 10 degree Celsius per minute on DSA purpose. So this is mainly based on the, that is thermal process. You can see here the picture is given. Okay, what are the main components of DSC? That is two, one sample holder is there and another one is reference holder is there. Okay, here you in the side, that is heater is already fitted, and this is the heat sink. Okay, so CPU is already connected with the DSC apparatus. One amplifier, two amplifier also required. Okay, these are the heat register and thermoscope, and one software and one, that means computer, is also required for the calculating and storing of the data analysis. Okay, so here one more important factor, that is temperature recording. Okay, main factor is that temperature. Okay, so what we can detect after interaction in DSC. Okay, by DSC what we can that means detect, that is elimination of endothermic peak, then any new peak appeared, change in melting point, oblique peak temperature, change in peak shape, height and width, change in area of the peak or enthalpy. Okay, so that means we have already validated some endothermic peaks of every, that means excipients or APIs. Okay, like paracetamol, we have already noted in which point, in which temperature the endothermic peak will come. If we mix two ingredients, that means one excipients and one API, if we add two ingredients in the same proportion, that is 1:1, and if we are going for the DSC study, if the endothermic peak of each and every compound is not in the proper position that is already validated, that means we can calculate it that some interactions maybe occur. Okay, so these are the main 5 points we can conclude by after detection of DS. Next come with the DTA, that is differential thermal analysis. Okay, so in this technique is useful to investigation solid state interaction of detection of eutectics. So in case of eutectic powders means it is not compatible with the normal environment. That means when you are put eutectic compounds in a normal temperature, it becomes melt. Okay, so it should be kept in the very low temperature. Okay, so DTA is required. We use DTA for the detection of eutectic compounds. Next, in this change in temperature between test sample and reference material is measured under control and ideal condition. So I have already mentioned that what are the conditions. Mainly the temperature is the main factor for the eutectic compounds. So this difference temperature is plotted against the time or temperature. Okay. So interaction can be identified by comparing DTA curve obtained from the test sample with those of reference material. So these are the main points for the determination of thermal analysis. Okay. So if any interaction occur, that means thermogram, that is called DTA curve of a mixture show appearance of or disappearance of one or more peaks corresponding to those of the components. That means you can see more than one peak, one or two peak, or less than two peak. Okay, it will be appearance or disappearance. Okay, it will be the one number of the peak is become, that means vary. Okay, the thermogram of mixture shows same patterns corresponding to the dose of the individual components. Okay, so if not interaction occurs, so it shows what activity, that means the thermogram, that means DTA curve mixture shows same patterns corresponding to those of the individual compounds. Okay, so this is about the DTA, that means differential analysis, thermal analysis. I have give the short description of the DTA analysis. So next, SIC, that is, so SIC is useful for potentious drug and excipients. So those who have potent drug, that SIC, okay, that means self-interactive chromatography we are using for SIC for the potent drugs, okay, drug and excipients. So what is the principle for SIC? That is for different mobile phases, different excipients. Okay, different mobile phases means different excipients that injected drug have different interactions, may be repulsive or attractive. With the stationary phase or drug leads to shift to in the retention time. Okay, so this is the principle. That means some that means for the different mobile phases we are using, that means different mobile phases means different excipients. We are using excipients as in mobile phases. Okay, the injected drug have different interactions. Okay, that means it may have the repulsive interaction or it may have the attractive interaction, okay, with the stationary phase. And by calculating the shift in retention time, we can calculate the what is the main property of the particular drug and excipients. Okay, so this is the principle for the self-interactive chromatography. So what is the method? How can you calculate, determine? So it is a modified type of affinity chromatography where drug is made immobilized at a stationary phase and solution to be tested. That is, solution is the excipients act as a mobile phase. Okay. So measure the retention time and compare with the non-retain maker, we can conclude the drug interaction of the drug and excipients. Okay, so this is the process of self-interactive chromatography. So you can see in this picture, these are the, that means graphs you can see. Okay, When interaction is repulsive, a sharper peak is obtained at the shorter retention time. Okay. When interaction is retentive, you can conclude that that means interaction is repulsive, or that means attractive. Okay. So if the shorter peaker time is occurs, okay, sharp that means peak is occurred, that means we can calculate it that in shorter retention time the repulsive interaction happens. Okay. Next, when not net interaction between the immobilized drug, that means RT, dated volume of codon. Okay, you can see, you can conclude from this picture. In next picture, figure C, that is when attractive interactions, it will have a longer retention time with wide peak. So from this study, we can calculate that self-interactive chromatography. We can calculate by the graph. We can calculate it by the graph of the particle. Okay, graph of after chromatography. We can calculate what is the main properties of the drug excipients compatibility study. Next come with the TLC and HPTLC. Okay, so TLC is generally used to conformative test of compatibility after performing DSC. Okay, so after performing DSC, we can conclude it, that is the confirmatory test, that is the last test, that we can conclude that they have no interactions, or they have, if they have the interaction, we can conclude it, we can confirm the interactions. So DSC, because it is sample undergo negligible thermal changes, it will difficult to detect by thermal method. Okay, so some that means molecules, some chemicals have, they have the less thermal, they have less tendency to active in thermal properties. Okay, thermally they are less active. We are putting the excipients with the DSC apparatus, they have shows the less response. For that particular case, we are using TLC to confirm it as a confirmatory test. Okay. So what is the method? Method is that one is the mobile phase, another one is the stationary phase. So stationary phase consists the powder, that is silica, alumina, polyamide, and cellulose, etc. These are the main the stationary phase. Next, adhere on the glass, plastic, or metal plate. So students, you can, I know that you can already perform the TLC, that means study. Okay, that means experiment you have already performed. So one is the main component of the TLC is the mobile phase and the stationary phase. So here we are using stationary phases, silica gel, alumina, and polyamide. So solution of the drug, that is excipients and drug, we have mixed the solution of excipients and drug. So excipients mixture are prepared and spotted on the same vaseline at the end of the plate. Okay, the plate is then placed upright in the closed chamber containing the solvent, which continues the mobile phase. Okay, any changes in the chromatograph, such as an appearance or new spot or a change in RF values, the component is indetective of an interaction. So this is about the TLC and all the simple analytical method for the preparation of, that means not the preparation, that is compatibility study of the drug excipients. Okay, so this is all. Thank you.